Hello, and welcome once again for another episode of Profiles in American Funded Research. I'm excited for our guest today, Sarah Sheffield. Sarah went to the University of North Carolina at Chapel Hill to get a bachelor's of science, and then got a master's degree at Auburn University, got her PhD at the University of Tennessee, and currently is at Binghamton University as an assistant professor. Is that everything correct there?
Yep, I was an assistant professor for about 5 or 6 years at the University of South Florida before coming here.
Okay.
Mm.
And I invited you on today to discuss this project that you have with other people as well. In fact, a previous guest what is involved in some way in this project, I understand. But the project's title is Paleozoic Echinoderms as Model Systems for the Study of Evolutionary Modes.
Mhm.
Would you be able to give us just a brief overview of what that project is about?
Yeah, so echinoderms are the fossil group that includes modern animals like sea stars and sea cucumbers, sea urchins, so things that are still alive in the ocean today. And their diversity is greatly reduced. We have 5 major groups of them today, like crinoids and the ones I just mentioned. But throughout geologic time, we've actually had closer to 30 of those groups. And the groups that used to live, most of them lived in a time called the Paleozoic, so about 545 to 251 million years ago, just about. And they were extremely diverse, not just in taxonomy, but in their morphological shapes, how they lived, etc. So they were extremely experimental. They had all kinds of different body forms. And the Paleozoic was a time on Earth where we had a lot of climate change. We had really high highs, really low lows, so extreme glaciation points, times where all the ice caps on Earth melted and it was extreme greenhouse environments. And echinoderms lived through it all. And we can actually see their morphology changing throughout these times. So we are testing hypotheses of how echinoderms would have evolved in response to these climate change events. Those that survived, those that didn't, why would they have? Why would they not have? Are there trends that we can find? And one of the ways that we're doing that is we're taking the evolutionary relationships of these organisms. We're building evolutionary trees using the fossil record and different computer programs, excuse me. We're then looking at the biogeography, so where they moved across these events, and then understanding how those would have tied to the changing paleoecology and the changing of their morphology through a number of different ways.
And for viewers that might not be familiar with some of the terminology, can I just ask you to briefly explain what taxonomy
Ye-
means and morphology?
Of course, so morphology is shape. So they're changing shape. And taxonomy is it started with Linnaeus. So naming of animals and plants and things like that. So Homo sapiens is the taxonomic name for humans that are alive today. So that's our genus and species, how we name our fossils.
Mhm. Thank you very much. So, one thing that I was curious about, I guess I was pronouncing it wrong. It's echinoderms.
That's okay.
Yeah. Um, how motile are they? Because when I think of a starfish, I just sort of think of it as floating around in the ocean. Being taken by currents mostly. But how motile are they?
That's a great question. So the answer is it depends. And so some organisms, sea stars, starfish, they're more mobile than other groups of echinoderms. They use their little tube feet. So if you flip a starfish upside down, you see those little things, little suction cups moving around. Those are its tube feet. But that's in its adult form. In its larval form, all echinoderms that we know of started in a larval form. And those larvae are free swimming and they're very tiny, of course, they're larvae, and they're able to be transported by different oceanic currents, things like gyres. If you're familiar with Finding Nemo, that turtle that was getting sucked into those things and floating around, that's a gyre. So echinoderms will use those in their larval state. That's how they're actually spreading their range. So, if you're considering the fact that we're talking over millions of years for a lot of these events, even a couple weeks and, a couple thousand meters of distance really can add up to global distributions. So that's how they're getting around, is mostly in that larval stage. Once they're adults, they're more sessile, meaning they don't really move that much.
And how long do they live?
That is also a great question. So the answer to that, I'm so sorry, again, is it really depends. Some can live for quite a few years, some not nearly as long. It really depends on the group of organism. If we're talking in the fossil record, we don't genuinely have a great handle on that at this point, mostly because one, there's not a lot of growth studies that we can do in the fossil record because we don't always have enough fossils to do that data. And two, with echinoderms, it's a little harder. We don't always have the data to show what age marker. So, for example, if we're looking at a human fossil, we can identify the age it is based on the eruption of the molars or possibly hip position and things like that. We can tell if it went through puberty or it didn't. We can tell by the fusing of the bones if it's a child or an adult. We don't always have that type of hint with echinoderms. Some have them, some don't. So it's a little bit harder to tell. We don't necessarily think it would be any different than organisms today, but we can't necessarily always say that. Yeah, we don't have enough evidence to really say that.
Yeah, definitely. I can understand there's a lot of uncertainty that goes into this kind of
Mhm.
research. So how are you able to then, because of that uncertainty, how are you able to track the evolution of these species?
Mhm. So, taxonomists, people who study the different species of different groups, we are highly trained specialists. So, I've been studying taxonomy of echinoderms since 2011 or so. So, oof, that number keeps getting like exponentially larger every time I think about that. Wow. Ah! But I've been studying this for nearly 12 to 15 years or so. And so we are trained to look at the smallest of details to really identify these different fossils. So we use that training to be able to better understand how different things can be before we can call them a different species. And there's a number of different tests and visualization techniques that we can use to better quantify that. So it's not just my opinion, of course, saying I think this is a different fossil, because we don't want that. We want hypothesis testing, we want rigorous, repeatable methods. Um, but in general, we spend a lot of time looking at these fossils, and we don't try to. One of the things that we really try to avoid is hinging our opinions on something that could be related to growth. So, for example, with echinoderms, we'll have. Can you imagine with me the five-ishness that a sea star has? It has the five limbs. So the two that stick out to the side, kind of like if I was a starfish, I look like a normal human. For those of you who can't see me, I have a head, two arms, two legs. If you stood straight out with your arms and legs out, kind of like you were about to do a cartwheel, you would have the two arms. Those actually erupt at about the same time as the legs do. They come out about the same. And the one that would be my head comes out last. So in juvenile fossils, every once in a while, we don't usually find these too terribly often, but if we found it before that stage, I could definitely say that is juvenile. But then I wouldn't go and write an article stating this species only has four limbs or ambulacra, which is what we would call it. I would know that that's based on a juvenile trait, right? So we try to use the best we can to not hinge our opinions on those juvenile characteristics or things that might be influenced by the environment. So, for example, we also have characters, so parts of the morphology, so like the way your head is shaped, the way your mouth is shaped, things like that. Some of those can actually be controlled very strongly by just the environment you're in, not necessarily your genetics. That's what we call enophenotypic plasticity. So, eco meaning ecology, phenotype, the way you look, and then plasticity meaning moldable or changeable. So, really long word, but it actually breaks down quite nicely. Try saying that 5 times fast on a podcast, though. Uh, so, ecophenotypic plasticity can be like a lot of you might see this in freshwater streams. Freshwater clams will often have different shaped shells based on how fast the water is moving or how clear the water is. And that's not a genetic thing. So we wouldn't use that to understand the differentiation between species. We would know that that is something that can be affected. So, for example, in echinoderms, oftentimes we'll find the holdfast, which is like the stem of many of them. Many of them are stemmed. There's 2 different types. There's a holdfast and a true stem. They're slightly different, but some of them can actually have the end curved. They were actually growing on something. So I wouldn't say, "Oh, this species has a curved holdfast." I would know that it's because it grew on something like another shell. Does that kinda make sense?
Yes.
So in general, it just takes many, 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 many years of studying these fossils to really truly understand them. And of course, I'm not the only person in echinoderm taxonomy. So another important part of it is other people study these. So they have their opinions, and we write our papers and we argue about it in a professional way, of course, to strengthen or change our interpretations.
Yeah, I'm familiar with arguments among communities. We have arguments in my community as well. Um,
Healthy arguments, of
yes,
course.
yes, healthy argument in scientific papers actually. Um, so um, correct me if I'm wrong, but I think that there's this term that I saw in maybe one of your papers or something that's disarticulation. And my understanding
Mhm.
is that is the spreading out of body parts, right? Over is that correct?
So, disarticulation is something that happens to fossils. Well, let me rephrase, to organisms before they become fossils. So, our bodies are held together by ligaments and joints, so soft tissue. So, our bones and shells and teeth are really good at making fossils, much better than other things. That's why we typically only find what we call hard parts, bones, shells, teeth, things like that, but ligaments hold those joints together. So after an organism dies, the soft tissue starts to rot. So the bones get separated. And if you think about a human, we have somewhere between 206 to 210 bones, depending on if you're a juvenile or an adult. Echinoderms can have over 1 million body parts.
Oh wow.
Yes. No, when you look at a sea star, if you look at all those are all individual pieces, kind of like your humor, your arm bone, or your leg bone. Those are all individual pieces that are held together by soft tissue. So disarticulation is what happens when an organism dies and the body starts to break down. Pieces of it can be separated. This is why finding fossils is such an incredible gift from the universe, such an incredible gift because they often fall apart very easily. So the majority of the fossil record doesn't necessarily have complete preservation. We can still tell what's there, right? Because a lot of the features will still be there, but not the entire body will necessarily always be there, or specific parts of it will be. Or to have gotten them to be complete, they would likely have been buried alive, like in a storm event or something like that. It's the best way to make sure that we have fossils that preserve whole is if there was a storm event that just kind of flattened them out.
Uh, so I think roughly my understanding was correct there then. And my question then to you, and you sort of hit a little bit on that already, but was so in the case where you have disarticulation, where you don't have the entire specimen, are you able to still use the pieces when you can kind of tell what they are? This is a
Mhm,
starfish as an example. Um, are you able to then use those pieces to still help you when you're studying things like the change in climate and thing going back
mhm.
in time?
So a lot of the times, just because, and I'll give you an example. So a lot of our echinoderms in the past had arms. So they would be little plates, not like arms like you and I have arms, but the little plates that stack on top of each other. And are you familiar with the wacky inflatable waving tube men that like you can see
Yes.
from outside? Arms, they're not a direct analogy here, but they're very similar the way they wave up in the air like that. And for any other echinoderm workers listening, please forgive me for this explanation. But they're kind of similar to that, and they have typically 5 of them or multiples of 5. Echinoderms love the number 5, so they are not pentaradial in symmetry. They have what we call a 5-ish to them. They're not truly pentaradial, otherwise they would have like
They're
5
close.
every, but they're,
Yeah, okay.
yeah. So,
Nature's not perfect.
yeah. And, but if things were perfect, I think echinoderms would be at the top of the list, but I'm biased. So, those arms are extremely delicate, and the things holding them together, those ligaments, are the most delicate typically of all the other plates. So, most of my data set does not include those arms. I usually don't have them, but we do have scars. So, if we were to look at a human body, I know where on the femur the muscle's attached to because there's a scar left behind. I don't need to see the muscle to know where it was because that scar is left there. It's the same with the echinoderms. There's small scars where the arms would be attached to the rest of the body. We also sometimes will find a few of the arm plates, maybe not all, but a few. And if we're talking about organisms maybe that we have many fossils for, like thousands of specimens, chances are we might be able to find one or two. So just because we don't always have it, it doesn't necessarily prevent us from making the same judgment calls or the same scientific interpretations. It may mean that we aren't able to always describe things in as clear detail as we would like, but that's the nature of science, right? So, of course, as scientists, I would never try to describe something that I cannot see or that I cannot confirm for myself. That's okay. I can go back and I can revise later. So, we recently found Don Bissett, who is a avocational paleontologist with the Dry Dredgers Fossil Club in Ohio, which is one of the most famous fossil clubs in the United States. They're awesome. He found one of the only examples of a specific group that I work on with arms, and it has half of an arm maybe. We're not really entirely sure. I would guess half, but I wouldn't be willing to put that in a paper unless I could confirm it more clearly. And so that really helped us because arms, they're not going to change too terribly much. We're not going to see them, you know, there's only so much change in the shape that we can do here. So we can actually see how it looks and we can start to extrapolate how those arms would have looked on very closely related organisms as well to kind of help us picture how they would have looked and how they would have fed.
So you just mentioned something that I hadn't heard of before, fossil club. So
Mm,
is this like is this only a group of scientists, or is this like also like a citizen science type thing? I'm curious about that now.
Fossil clubs are phenomenal. So if you've never heard of them, I hope you can look into this and maybe you'll get lucky and there'll be one near you. Fossil clubs are clubs for people who love fossils. So these are people who are maybe are career paleontologists, but more often than not, most fossil clubs are staffed by non-career paleontologists. So what we typically call avocational. And these folks, they love paleontology down to their bones, pun not intended there. They go out every weekend, they scout out the best places, they know the local geology like the back of their hand. So I get to work with them and because I am paleontology is my job, which means I don't often have, weekends to spend on this. I have to do other parts of my life, spend time with my family and things like that. And so they'll go out and they'll find fossils and they will give them to me and we will work together on it. So, avocational scientists, citizen scientists have been working with me for a long time. They are on papers that I publish as a gratitude for finding the fossils and making that science possible in the first place. But fossil clubs are amazing. They're so much fun and they do a lot to further paleontological science across the world.
Yeah, I'm a very big fan of any kind of citizen science because those people tend to be really passionate about what they're doing, and as you said, spend time on their weekends or whatever doing so, like, to advance science without really getting paid for it is sort
It's
of an
beautiful. I love
yeah, it's an amazing thing. Um, so the the next question that I had for you is, why did you choose? I mean, I guess because partially the answer is because you're an expert in them, but why did you choose echinoderms in this case for studying these specific kinds of things like change in climate and whatnot in the past?
Yeah, that's a great question. So you're right. I was about to say I chose it because I am, an echinoderm taxonomist by training, but it does go beyond that, and that the echinoderms have not been well studied with a lot of these newer techniques for analyzing changes in climate and things. And part of that is because, as a whole, the echinoderm community is a little smaller. We don't have a whole lot of us. And a lot of this research is relatively new. We're starting to learn more and more and more. And we're starting to see a lot of different analyses really starting to take off. But echinoderms are incredibly picky. So, echinoderms today, to be clear, echinoderms today are very picky. They like their salty water a very specific way. They like their clearness of their water a very specific way. And what that means is if that's true now, there's something called the principle of uniformitarianism. Very long word to just say we assume the major processes that govern the science around us are similar today as they were in the past. So, for example, if we're thinking about dinosaurs, I can't tell you with definitive certainty that gravity operated the exact same way and dinosaurs were walking on the ground and not instead of floating in the air. But it'd be really silly to assume that they weren't, right? Gravity has been in effect in a similar way on Earth for 4.56 billion years, the age of the Earth. So echinoderms are very picky with their where they live today. It's likely that they were similarly picky in the past. And what that means is we can use that information. We can use how they respond to different things like salinity changes, temperature, etc. We can use that to build a roadmap to the past, essentially, to understand, okay, if that's the case, we can start to test hypotheses of if the oceans got really warm, I suspect that echinoderms may have had a global migration to cooler waters, essentially. And then I can test that hypothesis. And that allows me then to take what I've learned about long-term trends of echinoderms and say, okay, well, if I know echinoderms today are picky about where they want to live and the environment they're in, what might they do in an ocean that is warming? So I can use both the present and the past to better understand each other, which is the beauty of geology. It really is. We can take what we've learned and apply it to the future or the present, while also taking a look at the present and saying, okay, well, if I know this now, what might have been true a couple million years ago?
And when you're trying to make so obviously, if you want to kind of track things like evolution and whatnot,
Mhm.
you have to have some idea of time. So how do you get the dating? How do you do the dating
Yeah.
of these different fossils?
A great question. So, with this, I'll explain the first part of it, which is building an evolutionary tree. So, this takes all of the characters in morphology, so like the shape of the mouth and things like that we've talked about, and we build it into a very large database. And we take different algorithms to rearrange all of the possibilities of how they're related to one another until we find one that is best supported by the data. And there's a number of different ways that we do this, but I don't want to get too in the weeds on that yet, unless you're really interested later. But for the dating, once we have our evolutionary tree, we will take what's known as the first occurrence date and the last occurrence date of every fossil, and we'll input that into our data set. So the first occurrence and the last occurrence, that's not saying it's the first ever evolved of that species. We can't know that in the scientific record. We can never say we know exactly when the last T Rex took its last gasping breath as the meteorite came crashing down. We can't know that for sure. Highly dramatized for entertainment here. But we can know when the last recorded one was. So we always use that phrasing to be very cautious that we don't overinterpret our data to say that, we know for sure exactly that endpoint there. But one of the ways that we know these dates is through a combination of relative and absolute dating. So absolute dating can be something like me, Sarah Sheffield. I am 35 years old, 35 years old. Wow. Okay. It's Monday. I am 35 years old. That is absolute dating. Relative dating would be my sister is younger than me. You don't know how old my sister is, but you have a reference point to know that she cannot be older than 35. So with the fossil record, we have different species that evolved and had global lineages, or they're found globally. And we know that they lived at around the same time. So we can essentially say, okay, all of you lived at the same time. All of you of a different species, you lived at the same time. We can know that species one is older than species two. And with that, we have thousands of fossils that are really great at this. They're called index fossils. And the process of making these charts of how old things are, that's called biostratigraphy. Bio meaning life, stratigraphy meaning layers. So using the fossils to build layers to understand the different times. So we can use that to build these really complex and detailed time charts that never actually have any numbers associated with them. That's relative dating can be extremely precise, but we do also have absolute dating, and we like to take the two and make sure that we have as many lines of evidence as possible. And absolute and relative is extremely powerful. So absolute dating, we use radiometric dating. There's a really common misconception that radiometric dating means carbon dating. Carbon dating is only one type of radiometric dating. Carbon 14 specifically decays into nitrogen 14, has a half life of 5,760 years, I believe, give or take a couple years. So it has a fairly short half life. We have many, many unstable elements that decay with a much longer half life. So, for example, there is a system called rubidium strontium. So, rubidium decays into strontium, and that has a half-life of quite a few 100 million to billion years. So we can use both of those lines of evidence to build a really strong case for how old these fossils are.
Yeah, I know that, there's I think it's like uranium-235 or something like that, that
Mhm,
actually has a half-life the length of, Earth's age, essentially, 4.5 billion years or something like that. So
mhm,
there's definitely different ones that you can use to your advantage,
mhm.
for exact dating, but or not exact, but you know what I mean. The
Absolutely, yeah. Mhm,
it is it so now because you're also going to be dealing with different, uh, I assume a pretty large time scale because you were talking about a couple 100 million years ago
mhm,
that you were looking at. How do you account for things like we know, for instance, that the plates are shifting in time, right?
mhm.
So how do you account for that kind of those kinds of um, changes that are happening over time, over perhaps a large time scale? But when you're then trying to make these inferences,
That is an amazing question. And it is one that, actually that's so funny. My master's student is writing her thesis right now, and that's actually something she's trying to write in there to better explain some of the, like, assumptions and biases that are inherent to paleontology. So, plate tectonics. What happens every 180 million years or so, give or take a couple 10 some odd million years, we have a recycling of our oceanic seafloor. So the plates are moving. We have two types of crust, oceanic and continental crust. Continental crust is not very dense, so it never subducts. When two plates come together, it either rides over that oceanic plate as the oceanic plate subducts, meaning it goes down into the mantle, or continental plates can make mountains, like the Himalayas are two continental plates bashing up against each other in a race to see who's gonna get to the top first, essentially. Um, so with that, I'm dealing with fossils that are hundreds of millions of years old, meaning I have none of that data from the oceanic seafloor. However, that's not the only place we find oceans. So, the continent of the United States is actually larger than it appears today. Many parts of it are underwater. So, for example, Florida is about, I couldn't give you a great percentage without being very wrong, but Florida is actually a lot larger than it looks like today. Water has flooded the crust that edges. It's called the continental shelf. The same thing with the United Kingdom and France. The land that separates them, the ocean that separates them, is actually continental shelf that's been flooded. So if we just lower those sea levels a little bit, you could take a little walk there instead of the train, right? Or pretty long walk, actually. So what we have isn't data from the deep ocean. That gets recycled back into the mantle of the Earth through the process of plate tectonics. Every 180 to 200 some odd million years, there's like really old deposits in like the Mediterranean that are closer to 200 million years. Really cool. Those are called ophiolites. Look them up later. They're really fascinating. And so what that means is my data set is almost exclusively what lived on continental seas. So, through time, we've had sea levels rise and sea levels fall. That means sea levels have been much higher and much lower than they currently are today. And the Ordovician, as a whole, they were much higher. So much of the United States was underwater, as was the UK, as was Sweden, Norway, Estonia, places like that. Huge swaths of Africa, South America, Australia, they were underwater. So, when that water recedes, we have the fossil record left behind. That's where we're finding those fossils. That does mean that it is inherently unknowable to me what happened in a vast majority of the deeper oceans. So, echinoderms today live in both shallow and deep oceans. I don't have a great handle on who lived in the deeper oceans hundreds of millions of years ago. It is one of the parts of science that is unknowable. We can learn little bits Sometimes we have some preserved oceanic crust. If you imagine with me these plates when they're scraping together, I always kind of like to explain it to my students. It's kind of like the dinner dandruff that's left behind when you're done doing dishes. It's like the stuff that gets scraped up as the plates are scraping together. So sometimes we'll see some stuff there, like left behind. It really depends. There's usually not a lot, but we can still see it. But that means that we are missing large parts of that data. And that's something that it's not a negative, right? All science has bias, all science has unknowables to them. We just always have to be able to recognize it and make sure we understand that I can't extrapolate further than the information that I have. So,
Can that be? I'm curious now because you were saying about the fossil record, being on land, but I there's also some drilling, right, that happens that you could actually go out
okay.
into the. Are you able to use that to fill in some of those maybe mysteries that come from the current fossil record? Or
We it can happen. So echinoderms are typically not going to be very well preserved in these areas. So a lot of the times with the deep ocean drilling program, because I think one of your interviewees, Dr. Adrian Lamb, probably talked about this a little bit. Some of that data is too deep in the ocean. So there's something called the CCD, and the CCD, the calcium compensation depth, is a point at which in the ocean where water is so acidic it dissolves calcite. Calcite's the mineral that echinoderms are made out of, and that's CaCO3. And you can actually take a great look at Florida right now to understand how that actually works. Florida is built out of limestone, which is the rock that calcite makes up. And too much acid rain leaves us with lots of sinkholes in Florida, which we can see very evident. Those are very clear there. So that's how that process works. But on a micro scale with our individual fossils, when the water's too acidic in the ocean, so the deeper we go in the ocean, the colder the water gets. The colder the water is, the easier it is for essentially to hold more acidity in it. You can do this example actually with seltzer waters, cold and warm seltzer waters. You can actually get different pH readings off of them. That's a really great way to do it. But it also holds in more CO2 gas, which is why it's more acidic. So past that point, most echinoderms will actually dissolve. They can't live down there either, but their bodies will also dissolve as well. So when we're under specific depths, fossils that are calcitic don't make it. We have things that are made of other types of things like silica, SiO2, kind of like your opal rings. Those are also silica, things like that. They can survive, but calcitic organisms can't. In shallower waters, so if we're doing things like right off the coast of things, we can see them. But typically we're not able to get so far back in time where we haven't really gotten back too far to older oceanic crust. So, for example, one thing to remember is even though we've been looking at the seafloor for decades, we've only explored about 4 or 5% of it now. The ocean is vast, and we know more about the moon than we do about the deepest part of the ocean, which is always, I don't know, it's always one of those facts that just like makes me love science that much more to think about what we still don't know yet and what we can learn in the future if we fund science and make it a priority, of course. And so that's kind of, you might be able to find some, but echinoderms are probably not going to be the main players there, unfortunately.
now, but could you describe something that maybe you found surprising in over the course of your research that you about echinoderms that maybe you weren't expecting or that the evidence points to?
That's a great question. So, one thing I wasn't expecting is echinoderms don't play by the rules a lot of the times or what we expect them to. Echinoderms have very little respect for our knowledge of taxonomy and what we think should be happening, which is one of the reasons why I love them so much is that everything ends up being a huge surprise. So, I study a group, my specialty is within a specific group of echinoderms. They're called the blastozoans. And they're a stemmed echinoderm group. They're extinct. They died, the last ones died about 251 million years ago in a time called the Great Dying, because paleontologists are highly dramatic. A time on Earth where about 96% of life on Earth died from massive amounts of volcanism on Earth. Excuse me. And one of the things that blastozoans, we used to categorize them. And so this is kind of, again, going back to Linnaean taxonomy of like dogs, cats, those big groups, right? So carnivora was broken into the felines and the dog, canines, things like that. We defined blastozoans by the presence of specific types of respiratory structures. So one type of respiratory structure are two little holes punched into a specific, like a depression into part of the body, and gas exchange occurred across a thin film covering that depression, where we would have those two pores circulating oxygenated gas, and then we would have seawater flowing, or excuse me, gas exchange occurring across that boundary with the seawater and the body fluids. So very similar to how a lot of echinoderms will respire today. Some of them had slits inside the body where water is filtered in, and that gas exchange occurs across slits in the body. So it seems like a reasonable way to define them, right? Because they're really different from one another, these respiratory structures. They're what we call disparate, meaning their shapes are extremely different from one another. But what's really fascinating is echinoderms actually, we shouldn't define them by their respiratory structures at all. And the reason is once we started doing evolutionary analyses and we started putting in all this data to understand how they're related to one another, the respiratory structures actually re-evolve. So it's kind of similar concept to wings. Everything with wings isn't in the same group. So pterosaurs, like pterodactyls, or birds, insects, bats, wings re-evolve through time. That's what we call homoplasy. Homo meaning looking the same, plasy meaning plastic. So it just looks the same, but isn't really. That's how the Greek would kind of translate to that. So the respiratory structures continuously re-evolve. So the group I work on the most right now, because they're the weirdest, they're called diploporans. And if you imagine with me a potato with a stem and with holes punched all over the body, but make them super cute, right? Because they're precious. I promise. Google them. I will maybe convince you of this. Um, but these diploporans had these little two holes punched all over the body. But diploporans themselves, so organisms with a diplopore respiratory structure, meaning that double pore, two pores punched into the body, they actually show up 3 or 4 or 5 times throughout the history of echinoderms. So now that begs the question, why? Right? Why are they re-evolving? So if we go back to wings, why would wings re-evolve? Well, wings are awesome, right? Wings mean that you can get to different food resources. If an area isn't a drought, you can move yourself to another one. You can get access to the best of mates, all kinds of stuff. They have drawbacks. Wings are incredibly expensive metabolically. Birds have to eat a ton to be able to power themselves, but they have a lot of benefits as well. So it's the same with echinoderms. There must be something that is beneficial, right? So there must be some kind of environmental factor that allows for that re-evolution of these to keep coming up, right? So is it shallower water? Is it warmer water? Is it less or more acidic water? We actually still don't know. So this is some of the active research that my master's student, Elizabeth Altier, is looking at. She's actually going to be defending her thesis in a couple of weeks here. So there's some really interesting things that we're starting to understand. And in fact, some respiratory structures get lost entirely. So many echinoderms don't have them at all. And that was really surprising to me, but we're starting to see a trend in if you've lost your respiratory structures, typically your plates, the plates of the body, you can think of them just kind of similar to like a snail shell or something like that, the external part, not identical here, but the external part of the body, they get really, really thin. So gas exchange could likely occur across it. Some of them also have enlarged butts. And that's something we see in echinoderms like sea cucumbers today, where without respiratory structures, they often have larger butts. And you might be thinking, well, gee, Sarah, that relationship makes no sense. It actually does. Sea cucumbers can diffuse oxygen across their anuses. So we're thinking that maybe in the past, that enlarged anus would have allowed for more oxygen to diffuse if we've lost those respiratory structures as well. So we're starting to see a lot of patterns show up that is just, they're wild, right? Understanding these things is just every time we find one tiny piece of the puzzle, the other pieces just get moved away from us and we've got to start over with like new questions. But that's kind of the beauty of it,
Yeah,
right?
that's actually really interesting because I never would have thought that, they have no traditional respiratory system at all
Yeah.
as an option, right? But
Yeah.
evolution is amazing that way too. Uh,
Yeah, so for your fun fact for the day, there you go. Sea cucumbers can breathe out their butts a little bit.
um, so of course, this podcast is um, aimed at people understanding where their research funds are going. So I wanted to just ask, where is the money? Where is the money going for this project? You know, what is it going to labor? Is it going to
Mhm.
um, I don't know, purchasing fossils or whatnot? That these kinds of things.
Yeah, that's a great question because that is a lot of folks don't know, and I had no idea about how any of this worked, until I became a professor, really, like, even as a grad student. Like, a lot of this is kind of it's a mystery to so many of us. So most of the money for this grant actually goes towards paying people to do the research. So we visit museum collections, and museums allow people to come in and study for free. They don't charge for that, but of course we have to pay to travel there and things of that nature. I pay graduate students to help me with research, so I'm training graduate students to go out into the workforce, and that money provides them the ability to eat and live in an apartment, which is always really great. We love that. As they're learning and starting to publish their own
But they
research.
they don't
We
tend to
are
get paid
also
much more than like a McDonald's worker. I mean, if we're being honest, they don't get paid a ton, a full time.
typically typically it would be
Yeah,
less than most, McDonald's workers, yeah. I think that it is one of the biggest issues I have with the science how science works today is I think graduate students should be paid a living wage. I am very, very firm on that point, but, in reality today, many of them do not
but
make very
you and
much
I don't
money at
have
all.
control over that. That's part of the problem.
Unfortunately, my control is limited. When I have control, I make sure that my graduate students are paid as much as possible, but, so the money is also going towards, specific museum workshops, so we're actually teaching undergraduates museum skills. So as we move into the future, we don't have a lot of folks going into collections work and, like, maintaining the fossil or the collections where we have all of these fossils, and fossils require very specific conditions to be kept well and kept organized. We need specific humidity controls and things like that. But these fossils are our history. They're our collective history. They don't belong to me. They don't belong to you. They belong to everybody. The history of everything that's ever been on Earth. And if we lose that, there is no replacing it, right? We can't make new ones. They're just gone. So the stewarding of these collections is so incredibly important. And so a couple of years ago in Brazil, there was a horrible fire. I don't know if you remember it, but it burned part of the Natural History Museum there and we lost about 20% of the world's pterosaur fossils in an
You
instant.
know, I didn't realize. I
And
knew
that
about the fire, but I didn't realize that was
Mhm.
that devastating. Actually, yeah.
Yes, and I think that really it was one of the few moments that I really seen where it hit the public how devastating that loss actually was. They were like, "Well, what do you mean? Well, where do you see" because, as a kid, so many of us have that bonding moment of going to a museum for the first time, remembering that dinosaur, right? I remember it just as clearly as many others do. But when we lose those, that's it. We can't guarantee we'll find more, for sure. We can't know what's there. We may have found the only specimen that we'll know of that particular species. So having people there to care for those collections and to make sure that they're safely cared for is critical. So our funding, which is a really teeny tiny part of any kind of a budget, we are actually training students from across the United States in workshops once a year to learn how to do these skills and to help them build their resumes and different aspects of that so that they can go and get jobs in order to protect these collections moving forward. So the majority of this money is paying for research. It's all paying for research. There's no equipment for this particular grant, no computers or anything like that. We have all the equipment. It's paying for students to get an education and do the research. It's paying for my colleagues and I to do the research, and it's paying to train students. So,
and
yep,
and the necessary travel.
and the necessary travel.
Yes. All right. Well, I always have one last question that I like to ask, for my own personal benefit, which is, how did you get started in science? You know, if you could go back to a moment in high school or maybe before or whatever, what got you started on this path?
This is actually a really weird and wild tale. I really liked science as a kid. I always did. I don't know that I remember like a specific moment, but believe it or not, I was a giant nerd in high school. This might surprise you. Uh, you know. So I was the captain of the science team for many years, and I was also in leadership in the marching band. And I went to college thinking I was going to be a music teacher. I really struggled with math and science in middle and high school. I struggled extremely hard. I loved it, but I loved learning it in a way that I loved learning it on my own terms, essentially. Like in a math class, that was not my place to learn. I loved astronomy and learning about that, but like the actual academics of it, I really struggled. And so at the time, I said, I don't think I'm actually smart enough to be a scientist. That's just never going to happen. And I loved music, so I said, "I'm gonna be a high school band teacher." So I went to the University of North Carolina at Chapel Hill, and I enrolled as a music major. And pretty quickly into that first semester, my professors helped me realize that was not my path in life, was to be a musician. I really hated the competitive nature of it, and I did not wanna be locked in a basement practice room for 16 hours a day. Partial exaggeration there. But I just hated it all of a sudden. I was just so shocked, and I had no idea what to do at that point because, like, that's the only career I had really let myself consider. So I took a random elective class, just I dropped my music class, and I was like, "I don't know what I'm doing." So I took this random class called Prehistoric Life. And on the first day, there was a professor, Joe Carter, he has since retired, but he was speaking poetry of sorts about the earth and how it evolved and how humans have learned more about it and dinosaurs and things like that. And it was just, I remember sitting there transfixed and I was just like, oh my God, how do I be you when I grow up? This is amazing. The way he was speaking with just joy emanating from every pore in his body about paleontology. So I went up to him after class and I said, dude, I love this. Or I think I said something like Professor Carter because I was slightly more professional. All right. I said, "How do I be you?" And he said, "Oh, get a PhD in paleontology." And I was like, "Sold." Changed my major that day. My mom nearly had a heart attack. She'll tell you she didn't. She always believed in me, but she didn't. Uh, and so that passion is really what changed a lot for me because I still struggled in the classroom with learning math and science. Like, chemistry was the bane of my existence in college, and calculus, whew, I really struggled. I had lots of tutors. It turns out that I had a lot of undiagnosed learning disorders that I now know about, and it makes me a lot better at teaching my students because I can recognize the struggles that I had and see it in them and how I could have helped myself then. But that passion is what kept me going. The sheer love. Every time I learned something new about a fossil, it was just my mind was blown, and I was like, "Oh my god," and I'm boring everybody around me with like, "Did you know this? Did you know this? Look at this snail." And then, people are nodding politely or less politely saying, "Sarah, please stop talking to me." But, I got an opportunity to teach a class as an undergraduate. They had, a graduate student leave the program, and they were desperate need of someone to TA that class I had taken, Prehistoric Life, to teach the lab section. And at the time, I had no idea what I wanted really with paleontology. I just knew I had to be that person. And from that very moment of teaching that first class, I knew I had to be a professor. I had to teach. It was just the joy of introducing somebody to what I loved was just I couldn't put it into words. I really couldn't. It just my soul just felt like I knew where I had been, like it found its home. And there's never been a time I've changed. I gotten very lucky to have found that passion, but, I've taught about 2,300 students across 30 some odd college classes now, and I love it as much as I did the very first time I ever taught. So, it's just really great. I love being in science because I love being able to share what I know with anybody who will listen. Whether you're paying to sit in a classroom and you can't escape or, if you sit next to me on an airplane, I'll tell you all about it. So,
That's really fantastic. Well, thank you very much for joining me, and you know, I hope to see you again soon.
yeah, it was great to thank you for having me. It was great to meet you.